Welcome. Please be seated. Welcome everyone at this Studium Generale meeting that we have on the series on my microbes and me. Before I start, the general thing, please switch off your mobile phones so that we will not be disturbed during lecture and questioning. Before introducing the speaker or giving other information, I would like to introduce myself. I'm Jeroen, working as a researcher and lecturer at the University Medical Center here in Groningen. I only simply with a few microbes, but later on we'll hear about dozens of microbes in and on our body, and I only focus on a few of those. Of which we have been told previously. Some of you have been there, I hope, 2 weeks ago with Wil Hoekstra, who nicely introduced this topic, this series. And today we'll have more a focus on my microbes and me, and the reasoning why my microbes are on the first part of this title, and we are at the end of this. So the setting of this evening will be that we first have the lecture of 1 hour, thereafter will be half an hour of questioning, and you'll have a chance to get your question answered by the speaker, whom we'll introduce now. Jeroen Raes, from all the way from Belgium, who has a nice history over there, did his PhD in Ghent and a postdoc thereafter. Went to Germany for a postdoc, Heidelberg, came back to Belgium, and since then elaborated very astonishing research lines with his work both in Brussels, and he just mentioned to me, we just met half an hour ago, on his transfer to Leuven and have a group there. And then he also has a research or leads a research in a joint inter-university discipline on the application of the research on the biotechnology line. With that, I think I won't spend more words on this and give the full next hour to Jeroen.
Okay. Well, thank you very much. Oh, it's high up here. And as said, my name is Jeroen from Belgium. I run a lab that studies the microbiome. And today I'll explain a little bit more what that means for you. And when I talk about you, I don't mean you. I don't know whether somebody had said this before me, but there's 100,000 times more bacteria on and in your body right now than there's humans on Earth. There's 10 times more bacterial cells on and in your body than you have human cells. And that means there's 100 times more bacterial genes present and active. So if you look at it in terms of cells, you are outnumbered 10 to 1. You are not human. You are a walking bacterial colony. But that's good news. We had over coffee, we had a discussion about that people in the general audience still think of bacteria as the evil ones. Well, those trillions of bacteria that are in and on your body, they're the good ones. They're your friends. They help you digest your food, especially when we talk about the bacteria in your intestinal tract. They form a first line of protection against the bad ones, the pathogenic bacteria. They provide you with essential nutrients which you don't get from your food. And the disturbance of those natural bacteria that are on and in your body actually is more and more linked to human diseases. The bacteria in your gut are not uniformly distributed. As you can see here, you have different amounts of bacteria in the different parts of your intestinal tract. And the largest concentration of bacteria is in your colon, your dikke darm, as they say in Dutch. The lower part of your intestine contains about 1 to 2 kilos of bacteria. So we can think of that gut flora, as we speak, that gut flora as an additional organ that we carry around. That is more or less the size of our brains. We're not born with the gut flora. Babies are believed to be born sterile or quasi-sterile. There's still some discussion on that. But the first seeding of that ecosystem, and you'll hear me use the word ecosystem throughout this talk, the first seeding happens at birth. When a baby is born, he or she engulfs the first sets of bacteria from its gut flora from the mother, from the vaginal flora from the mother, the fecal flora of the mother, the skin flora of the mother, for example, during breastfeeding. And that's the nucleus, that first bit of bacteria, that's the nucleus of the future gut flora that the developing child will grow. And here you see an example of 14 different babies whose gut flora was monitored in that first year of age. And the different colors you see there are different types of bacteria, and I don't need to go into details to convince you that that first year is very chaotic, right? You see throughout the life of these babies that some bacteria go up and then they go down and others take over, and what is happening there is ecological warfare. It is bacteria fighting for their existence against each other to see who's going to be the boss, who's going to be dominating this ecosystem. Because bacteria are like us. They compete for resources, and the resources in this case is food, our food, what we ingest. And by, let's say, 3 to 5 years of age, the baby flora has become stable. The stabilization process starts when babies switch to solid foods. And then by 3 to 5 years old, a flora of a baby is sort of like an adult flora and has become stable. Now, that first period of life is crucial. I'll come back to it later, but that's when the flora is developed, and that's when the flora is established. And perturbations in that first period of life can have long-term effects. So how do we study the gut microbiome? So the last few years have seen an explosion in our field. And the reason for this explosion is the coming of new technologies that allow us to study this ecosystem in much more detail and with much higher throughput than before. So we start from these little blue pots. These are fecal containers. We start from fecal material from healthy volunteers, babies, or diseased individuals. And from this fecal material, I don't know whether you realize, but the majority of feces, stoelgang, is bacterial, right? There is some fiber in there, there's some waste product in there, but the large majority is bacteria. And so we extract these bacteria from that material, we extract the DNA of these bacteria, and that DNA is sent through machines like this, sequencers. And these are the machines that are also being used to decipher the genome of humans, right? You've heard about this, the human genome. We can find mutations that predict your chances of getting a certain disease. Well, we do the same with these bacteria. We extract all the DNA of all these bacteria that are in our intestinal tract. We determine their genomes, and from these genomes we can learn a lot. And what do we learn? We learn what microbes are there, and in what abundance. So we have that many of this type, that many of the other type. But we also learn by peering into their genomes, we learn what they can do. We learn what metabolic functions they can perform. We can learn whether they have resistance against certain antibiotics. We can learn whether they have virulence factors, for example, in the case of pathogens, whether they might become virulent and therefore start attacking our body. All of this out of a small stool sample. And then we can start comparing. I can take person's A's gut flora and the person B gut flora, let's say a person that is, a vegetarian versus a meat eater, right? And we can compare the gut microbiome. We can compare the composition of the microbiome. Do meat eaters have more or less of these and that bacteria than vegetarians? Yes or no? But we can also peer into the function. So can we find more genes or enzymes in these bacteria of meat eaters that are necessary for the degradation of meat protein? Here I've given you the two types of information. We can get phylogenetic information, who lives there, and we can get functional, what can they do. And this technology has created a gigantic hype and boost in our field, because suddenly we had a looking glass through which we could identify all these microbes that we could never see before. Because previously, what people had to do when they wanted to study microbes, they had to grow them. You have these little Petri dishes, which you might have seen last time, where you put a little bit of material on and you would see where bacterial colonies would grow, and these colonies you could pick up and you could study. But we don't know how to culture bacteria. We only know it for some. And there's a cliche in the field that says that only 1% of the bacteria that are out there, we actually know how to grow. So 99% of the bacteria on this planet, we have no idea how to grow. And only since recently we have been able to study them. Because previously we had to grow them to study them. Now with these technologies, we don't have to grow them. We just have to get their DNA. Right? And that's what we're doing now. And because of this gigantic shift, we were able to start big projects such as the Human Microbiome Project. And the Human Microbiome Project is sort of a follow-up of the Human Genome Project. For the last 20 years, people have been studying human genomes. And then the microbiologists came and they said, hey, there's that other genome that is also important. That genome of the bacteria that live in and on our body. And we should really, really look at that. And the funding agencies, they listened. And what happened was that all over the world, big initiatives started. And we had the big American project, the Human Microbiome Project, HMP, in which more than $100 million was spent. We had big ones in Europe, MetaHit, 31 million, and other ones in China, etc. And we've been involved in several of these studies, and there's some results that I would like to show you. So this was a first study in which we looked at what is the flora of a normal, healthy individual. We didn't know before what the flora was. We had an idea, but we never used these kind of high throughput technologies before to study this at this rate. And so in this case, we looked at 124 individuals. So basically more or less the number of people that are in this room right now. And we got samples from all of these individuals, and we sequenced the hell out of these samples, and we tried to investigate what is in there. What bacteria are in there? What genes do they have? What can they do? And so we came to a reference set of 3.3 million genes. And that's really nice, because now with that set, we can map everything we get from new data sets. For example, in disease, we can map onto that reference. We had the blueprint. We had the set of genes that were present in normal individuals. Well, these 124 individuals, of course. What we learned from that data set was that you share more or less 1/3 of the species with your neighbor. Right? So you have an overlap of about 1/3 in gut flora composition. We also learned that we found 1,500, let's say 2,000 species by now in the human gut flora, and we find, let's say, 200 species per individual. So you have in your intestine an ecosystem of bacteria that harbors 200 different species of bacteria. And that's actually quite simple for an environment. If you go to soil, so just outside, you take a shovel, you take a bit of soil, you'll see that you'll have thousands of species in a few grams of soil. So the gut flora is relatively simple to study, and that's of course nice for us. But we had that reference data set, we learned a bit, but then again, it was only a reference data set, and you want to move forward, you want to go to the interesting questions. So one of the interesting questions we asked ourselves immediately after that study was, okay, but how is the gut flora different from a person from Groningen to a professor from Brussels? Well, we didn't have people from Holland and Belgium, but we did have people from Denmark, France, Italy, and Japan. Very different nationalities, very different food habits. We were thinking, yeah, we're going to see big differences, right? We're going to learn something about the effect on your nationality of your nationality or your genetic background on your flora composition. And we were actually surprised that we didn't see that much difference. When you take a step back and you look at the species composition, you'll see always the same big species groups, we call them phyla or genera, always keep on coming back. Namely, bacteria called Firmicutes and bacteria called Bacteroides dominate our gut flora. And to a lesser extent, actinobacteria, which you can also find in soil, and proteobacteria. And much to our surprise, if we dug a bit deeper, we actually saw that the gut flora of, let's say, a Japanese individual could be more similar to that of a Danish individual than to another Japanese individual. So think of that. You have a Japanese society with people that are having more or less the same genetic background, they have more or less the same diet, and still you can have a gut flora in a Japanese individual that is much more similar to a Danish or a French or a Spanish individual than to other Japanese. We even had the situation where we had the gut flora of a father and a son having not clustering together, having very different gut floras and being more similar to other people in this study from different countries. And we did not expect that. And how we actually in the beginning we didn't even believe these results. We said there must be something wrong. I remember heroic discussions between me and the PhD students saying, no, this is wrong, go back, this is wrong. But the results kept on coming back. So we had to change our hypothesis. And the hypothesis became, well, there must be something else. And we called them enterotypes, gut types. So, because what we found was that everybody in our study always was clustering into one of three, let's say, corners of the ecosystem. We had one enterotype which we called the Bacteroides enterotype, and there were some people in there, Japanese, French. There was another one we called Prevotella. There was another one we called Ruminococcus. And they all were different people in these three different classes of gut flora composition. So, but what does that mean, right? We have, in the one enterotype, we see a dominance of this group called Bacteroides. I mean, you can forget about these names after the lecture, but it's just, we had one group dominating. In the other, we had another group dominating. And in the other, we had something was, well, not that well defined, but not the other two, right? And we called it Ruminococcus, because that was the group that was in. But it's not just one dominating bug. It was an ecosystem. It was a mixture of different bugs that interacted, right? If you have one group of bacteria there, you always have his friends around. And if you have another group of bacteria dominate, you have the other group of friends. You have social groups, social networks of bacteria that tend to coexist. And they form this optima in what I call ecosystem optima. And I'm going to go back and forth. And so you can think of this as constellations. So here's an example which is much more tangible. These are forests. Forests do what forests do. What do forests do? They take carbon dioxide and they turn it into oxygen. That's what forests do. That's their job, right? And all forests do that. All forests have that functionality. But you can have a tropical rainforest to the left, which contains a high biodiversity, many different species, and certain types of species. And then you can have, to the right, a temperate forest, like the ones we know here. They're both a forest, but they're very different. There's different types of plants, different types of trees, but still they have similar functions. And then you can have a third type of ecosystem, which is, for example, a bamboo forest, which is very different. You have a very low biodiversity. Basically you have one type of trees, bamboo trees. But still, that forest is a forest. And so that's how you should think of these enterotypes. You can have the same functionality, but formed through different ecosystem constellations, right? And these constellations are everywhere, because I'll be mainly talking about our gut flora, but we have floras all over our body. We have a skin flora, we have an oral flora, we have a nasopharyngeal flora, right? We have an armpit flora, a belly button flora, About half of us have a vaginal flora. Everybody has these floras. Everybody has bacterial ecosystems, constellations of bacteria living in and on our bodies. And they all form these networks of social bacteria. And we don't have much evidence, but we even think they talk to each other. We saw, when we did the analysis, we saw associations between bacteria in our mouth and bacteria in our guts. And so we think that there's even links between these different ecosystems, but we don't really know yet. So where do these enterotypes come from? In the study that we did, we didn't have much data to ask. But shortly after our study was published, there was a study done by an American group who looked at these enterotypes, but they had a study on diet. They had followed for the people's diet for over a year, and people had been noting what they've been eating, and so they could calculate what the percentage of protein was, the percentage of fiber, carbohydrate, and all of fat that these people have eaten. And at the end of the study, they looked at these people's gut flora, and they could associate, at least partly, the gut flora type to the diet that these people were having. But the conclusion was that it was the long-term dietary habits, and it was not individual food items. So it did not really matter whether you ate hamburger or sushi yesterday. What was more important was that what you ate on average, how much carbohydrate versus protein you ate, how much fat versus fiber you ate. That average long-term dietary habit, that seemed to be determining what enterotype you had in that particular study. And that's something that actually made sense, even in the context of the Japanese and the Danish, because everybody had a sort of rich Western diet. The Japanese fill in this diet in a different way. They get their protein from different sources, but they still get a lot of protein. And we fill it in different ways. And there are differences, but if only the relative amounts of these basic food groups matter, then it can be that a Japanese has a similar food habit than a French person. But of course, this was only one piece of the puzzle, because this is only one study in one American cohort. Like our study was only one study with a limited number of individuals. And so we're still looking for other things that could influence that gut flora composition. And we also learn things from looking at the gut flora of not humans, but for example from mice. And so we have done recently a study, and we looked at basically there's different strains of mice, right? So it's hard to compare to humans, but let's say we would have the Americans and the French and the Belgians and the Japanese. You would have they would be all mice, but they have a different genetic background. And what we did was we put mice together from different strains of mice. We put them together in a cage, and then again these same mice together in a cage, same types of mice in a cage, and we had multiple cages. And the reason why we did that was we wanted to figure out what determines the gut flora. Does the genetic background, what strain of mice you are, determines your gut flora, or is it your environment, right? The cage, what these mice live in, what bugs they get by interacting with each other. And the interesting part is when we looked at the data, the main determinant was neither of the two. It was something that looked like enterotypes. We had in these mice, we had two groups whose composition was very similar when you would compare it to two of the three human enterotypes. So also in mice, something like this was going on. But what was weird in this case, these mice, they were all eating the same food because they were in a lab environment. We gave them their food. There was no food difference between the mice. So what determined then this difference in composition? And so we went looking, and what we found was we used a marker for intestinal inflammation on staking. And we looked at, is there a difference in this marker? And the answer was yes. We saw that some mice who had just a little bit more inflammation in their intestine had a different enterotype than the ones that were not. And so now we're getting closer to the link between the gut flora and disease. Determining whether you have a small intestinal inflammation affects the bacteria that live in your gut. Well, in this case, in these different mice. So the enterotypes was not only driven by food, but also by host processes. Right? Now, when we then took these mice, and we had these two enterotypes, and we only looked at one of the two enterotypes. So we took away that inflammation signal. We did see the effect of genetics. We did say the different colors you see there on the left. So these are graphs that sort of visually represent how similar different mice are. Each of these different little dots there is a mouse, and the closer they are to each other, the more similar their gut flora is. Right? And you can see just as well as I can that all the yellow ones seem to be more similar to each other than to the others. Right? And that the blue ones tend to cluster together as well, and the green ones also sort of. Well, these colors are the different strains of mice. These are the Japanese and the French in the mouse study. So if you take away that enterotype signal, there seems to be an effect of the host genetic background. So your own genome seems to be having some effect on the bacteria that you carry around. So you as an individual has some effect on which bacteria you have, but not completely, because we can only explain 20 or 30% of the variance in these mice by the genotype of the mouse. So only 20% of the variation that we see in gut flora composition can be explained by the genome of these mice. And there was another thing that we saw. We saw that mice that were in the same cage, they also had similarities. If you put a bunch of mice together, their gut flora seems to be slowly synchronizing. Not completely, but a little bit. Well, explaining 30 or 40% of the variation, which is quite a lot. So the environment seems to influence the gut flora composition more than the genetic background. Now, there's one difference that I'm not mentioning here, well, I'm going to mention it now. There's a difference between men and mice. If you put mice together in a cage, they do a thing they call coprophagy. What means is they eat each other's fecal pellets. They eat each other's droppings. And so what they're doing is they're transplanting gut flora from one mouse to another by eating each other's fecal material. But this is a natural thing these mice do. Right? And it could even be a natural way of maintaining or maintaining variation in the gut flora population. We don't even know. But that's why we have 30 to 40% of the variation explained by mice putting together in the cage, because they synchronize their gut flora by transplanting gut flora among themselves. In any case, there's still 50% individual variation. So there's still 50% of that variation that makes every single mouse unique. You all have your unique gut flora composition to some extent as well. So you can tell that even from a very simple experiment like this, you end up with hundreds of questions, because it's food that determines your gut flora composition. It's inflammation, but also your genetics, and also, if you're a mouse with whom you're sharing your cage with, right? Think of this. Think of your oral gut flora when you go to bed with your partner tonight. So there's a lot of things influencing this gut, the bodies, the bacteria that live on and in our bodies. And so there's a lot more to be discovered. Another example of the effect of the genotype or the genome on the gut flora composition. This is a very technical, specific slide, but the main message I want to put here is, if you take mice and you knock out, what people call knockout, you switch off one of the genes of this mouse. People can do that. People do that to study what the role is of such a gene, and then if you switch it off, you see whether the mouse has a different hair color, and then you know that that gene is for hair color, right? So what happens here is you switch off the one gene in the mouse, in this case a gene called caspase-1, doesn't matter, and you see a difference in flora composition. So we're getting to the point that we can find out which individual genes in our own genome can affect the composition of our gut flora. And this is one example. There's many examples appearing in the literature these days. So, we have some idea on what influences our gut flora. In all honesty, I think this is just the tip of the iceberg. There's much, much more to come. But what can we learn? How is this important? How can we link this to our health? Well, the one thing that we do also, we try to learn something about our metabolism. So what happens when we digest our food? There's a bunch of enzymes, proteins that our body makes, that are in our intestine and that are used to digest the food, right? To break down the carbohydrates, to break down the fibers, to break down the fats to smaller and smaller and smaller pieces so that they can be taken up by the body and used as energy and as building blocks. But we're not alone doing this. Our gut bacteria are a big, big help. Because our gut bacteria, they can break down substances and especially fibers that we cannot break down. And so in order to study that, we work on methods to reconstruct that metabolism in our gut by looking at these genomes of these bacteria. And to give you an idea of why this is important in a clinical setting, I'm going to give you a story of a clinical study that we did. So this is a study in obese individuals. We had a cohort of 200, more or less 200 obese individuals, so they were all overweight. But within this cohort, we found two subgroups. One group of individuals which had a low biodiversity in their gut, which means that they had less different species in their gut than the other group. Right? So think of this as the bamboo forest and the rainforest. Right? What we saw was that the people with the low species diversity, they were also the people that were obese but not healthy in their obesity. They were people that were evolving towards diabetes. They were people that have what we call low-grade inflammation. They had a continuous inflammation in their body that was not heavy, but it was always there. And we know that this low-grade inflammation can be linked to the development of what people call comorbidities of obesity, of additional risk to get complications, to become diabetic, to get cardiovascular diseases from obesity. That was the low diversity group. The high diversity group, they did not have this phenotype. So you had people that were healthy and obese, and people that were unhealthy and obese. And the difference, and the big difference, was in their gut flora, in the diversity of their gut flora. Now, these two groups were also linked to two of the three enterotypes that we saw. The Bacteroides enterotype was the unhealthy, low richness, low diversity people. You had the Ruminococcus, the not really defined one. That was the high biodiversity, healthy individuals. And then if you look into the metabolism, we could dig down and we could look at the functions, what these bacteria could do. We saw that the gut flora of the low diversity individuals, it was a gut flora that was actually not healthy for these individuals. There was a gut flora that had a decrease in the production of compounds, in this case short-chain fatty acids. You can forget about that. Compounds that we know that the gut flora produces for us and that are important for our health. There was a decrease in that production, and there was actually an increase in production of compounds that were bad for us. Carcinogenic compounds, production of ammonia, production of nitrite, all kinds of things that you would not want your gut flora to produce too much. But still in that group we saw. Now, of course, I'm simplifying it a bit, and we had no proof of causality. The only thing what you could say is that these people with that low diversity gut flora, they were they seem to be less healthy. Their gut flora seem to be less healthy for them than the other gut flora. And this is an example in obesity, but there's actually more and more and more examples of these kind of things appearing in the literature. There's not a day that passes and we don't find papers being published about these and these diseases. In these and these diseases, people have a disturbed gut flora. And you can see it's all kinds of stuff. It goes from obesity to diabetes to Crohn's disease, asthma, arthritis, all kinds of different diseases. And all of them seem to be linked or at least associated to differences in gut flora composition. And it's a bit scary, right? There seems to be too much, right? How can all of these diseases be linked to our gut flora? Our gut flora must be very important. And it's not small diseases. This is a Belgian slide, but these are diseases that are major numbers of individuals in each and every country. So we can think of what we then call gut flora-based diagnostics. If in all of diseases it's really true that the gut flora is disturbed, well, can we then not look at the gut flora and try to detect those diseases and hopefully detect them before they arise? And this is, of course, very, very early days, but the answer is yes, we seem to be able to do this. In some of the major diseases, we can have very high predictive power by just looking at stool samples. So give me the stool sample of a diabetic person, I will tell you he's a diabetic person. Give me the stool sample of a Crohn's disease patient or a colitis patient, I can tell. And of course, it's not always important. I mean, for a diabetic person, you can also measure blood sugars, right? You don't need to look at gut flora. But if we can start looking into the future, if we can start predicting that somebody will become diabetic, then this is going to become a very important tool. So, I strongly believe in that we will be using that thing that we flush down the drain every day, or depends on your habits. By using this to tell your health status, right? So like when you would go to the doctor and the doctor would take a blood sample, well, I don't think it's going to be taking long before the doctor will also take a stool sample and use that to diagnose your health. But we have to be very careful here. Association is not causality. What I've shown you so far is association. People that have diabetes have a different gut flora. People that have Crohn's disease have a different gut flora. It does not mean that the different gut flora causes Crohn's or causes diabetes. It just shows that it's associated to it. But there is causality in this field. This is a case of mice where you had a mouse model of an obese mouse or a fat mouse. If you take the gut flora of this obese mouse, you put it into what we call a germ-free mouse, which is a mouse which has no gut flora. You put it in there, you transplant flora, you'll see that these mice become obese. I transplant the flora and I pass over the phenotype. I make mice obese by giving them the gut flora of an obese other mouse. So there is some causality. This would not happen if the gut flora was not at least partly causing this, right? And I'm not saying obesity is only caused by gut flora, because we all know that there's many reasons for obesity. There's a genetic family history, there's your food habits, but apparently also your gut flora seems to be affecting how you digest your food and how this can lead to obesity. Another example is diabetes, type 2 diabetes, so elderly diabetes, not the aangeboren variant. People that have type 2 diabetes, they have a slightly disturbed gut flora, and you can build these diagnostic markers, etc. We can give me a diabetic person's flora, I can tell you that he's diabetic. But and this was done here in the Netherlands and in Amsterdam, if you transplant flora of healthy individuals into, this is in human, not in mice, into diabetic persons, you'll see that these diabetic persons, their insulin sensitivity, which is a measure for their diabeticness, increases. So we can start thinking of treating individuals by flora transplant. This is in humans now, right? By changing the flora of an individual, you can start curing individuals. Of course, this is very early days. This is a very small experiment with a small number of individuals. But it just shows you that there is more to it. It's not pure association. And we do the same in other diseases. We are now looking in, for example, IBD, diseases like Crohn's disease or ulcerative colitis. We're working together with the clinicians in the hospital trying to see whether flora transplantation cannot be used as a cure for these individuals. It's a very, very cheap drug, right? You can get it everywhere. But of course, there's a lot of questions, right? Do they all work? Is your mother's better than your father's? Etc., etc. Right? There's a lot of research that needs to be done. It is not common clinical practice as we speak. It is very experimental at the moment. Okay, back to the babies. That early period of life is crucial in the development of the gut flora. And we know because of experiments or situations where the early period of life where the flora of the early period of life was affected. For example, there's a study in mice, again in mice, where young pups of mice were given what we call subclinical dosages of antibiotics. So they got antibiotics, but very, very little. Too little to be of real effect, right? Very small amounts. If you give pups, mice pups, these very small amounts of antibiotics, you'll see that later on in life, they'll have a disturbed gut flora, and they'll be higher, well, they'll be fatter, basically, right? So messing with the flora at very young age can affect the gut flora composition and the phenotype of these mice at later age. We also know, for example, from population studies, that the usage of antibiotics in very young age, again, we're talking about that period, baby first two years of age maybe, that if we see antibiotics usage in that period of life, we'll also see a higher amount of asthma and all kinds of these types of diseases, eczema, and all, at later age. So you mess with the flora at young age, you get problems at later age. And you also see this even, and we don't have any, how should I say, diseases linked to this yet, or, well, we don't know. But for example, the way babies are born makes a difference, right? Remember that this first inoculation of flora happens at the moment of birth, and people that have been present at the birth, they know what kind of messy situation that is. So there's lots that can be ingested. Well, if you do it, what happens a lot these days, the clean surgical way by cesarean, that process does not happen. These babies are not inoculated by their mother's flora. And what we see is that when they grow up, the two groups have very different flora composition. So we don't really know yet what the effect is on the health of these individuals, but we do know that their flora is different just because that first inoculation did not happen. And going back to antibiotics, this is examples of antibiotics usage in adults. When you give broad spectrum antibiotics to an adult individual, what you do is you wipe out the flora, right? You kill the bacteria in your intestinal tract. Maybe not all, but a large majority. And then you see that different people react differently to this over time. So you have some individuals that after 2-3 weeks of time, they actually they regain their original flora, and everything's fine. For some people this takes months. For some people this takes almost a year. For some people the original flora never comes back, and they have permanently altered their flora composition after one dosage of antibiotics. And we don't know, right? We don't know how bad this is, but we only can see that the effects of antibiotics usage on the gut flora composition can be very, very important, and that they are very different between different individuals. Now, I don't want to say that we need to abolish antibiotics, right? Don't get me wrong here. Antibiotics is very, very, very crucial, and they have ridden a great deal of infectious diseases in the Western world. The only thing that I'm saying here is that we learn that by using antibiotics, we have a lot of collateral damage to our gut flora, and we should seriously think of not prescribing too much antibiotics, and there's also other reasons to do that, and especially not in that very early stage of life. There's some recurring themes, as you've seen from the obesity example. Actually, that example comes back multiple times. We see that in very many diseases, the hallmark of a diseased flora is a low diversity flora. So if you lose biodiversity in your gut, that seems to be associated to a less healthy status. And if you tell this to an ecologist, again, this is ecology we're talking about, an ecologist will say, yes, of course. Because also in natural ecosystems, plants, animals, the ecosystems that have higher biodiversity, they are more what they call resilient against perturbation, right? They have more backup systems. And so a flora that has low biodiversity is maybe less stable, can be shaken, can be disturbed much easier. And so we see that in infants with, in this case, eczema, in people with Crohn's disease, in obese people, in etc., we always see this reduction of biodiversity. So that seems to be a theme here. So what is then linked to this biodiversity? So one thing could be, again, that the inflammation reduces the biodiversity in our gut flora. But there's other things, and these are just topical studies, because this is a very new upcoming field and we don't have much data here. The first is a study where people looked at the biodiversity in people's gut and the biodiversity in people's environments. And they saw a correlation. So people that lived in environments where there was lots of, in this case, plant and animal biodiversity, their biodiversity in their own gut was also higher. This can probably be explained by the fact that if you have higher plant and animal biodiversity, probably you also have more bacterial biodiversity in your environment, and so you exchange more bacteria and you see many more different types of bacteria, and therefore your own gut bacterial flora is also more diverse. But we don't know, it's just an association. The other example is flora in elderly individuals. So what we know is that when people get older, their flora starts to deteriorate. From, let's say, 65, 70 years old, your flora loses out on biodiversity again and becomes less stable. And these people have studied the food habits of these individuals, and they saw that actually the diversity of your food was correlated to the diversity of their flora. So people that have had at older age still a very varied diet with lots of different food ingredients had a much more biodiverse and thus probably healthier flora than people that tended to move towards a very limited diet. And that is something that happens with old age. People tend to eat narrower in diversity. But it seems that if you keep that diversity in your food habit, it actually is beneficial to your flora. And so there's still lots of questions that need to be asked. And we're still, there's so many things that we still don't know. And this is also why we in our lab have started a project where we are looking into, in this case, the Flemish population. So we're gathering stool samples from the whole of Flanders, and the press loves this. Professor has 5,000 pieces of shit in his freezer. But it is very, very interesting as well. And so we're gathering all over Flanders data to just answer all of these questions. What influences your gut flora? Is the flora from a person who lives in the countryside different from the person that lives in the city? Can we relate this to other kinds of things? And if you're interested in the study, you can have a look. At the moment, we're not open for Dutch participants yet, but that might come. And so this is the field. This also becomes, a logistical challenge, right? Collecting stool samples all over Belgium, it also requires a lot of logistics, because you have to get all these samples and put them in your lab, etc., etc. And we've been very busy the last few years to manage this. But before you is a very happy professor standing, because we're getting hundreds of samples coming in every week now, and so we'll have a lot to do in the coming years, and that's what they look like. So, my conclusions. Take care of your friends. We start to know more and more about what a healthy flora is and what not. But at the same time, I have to admit that in many cases I still don't know what a healthy flora really should look like. And that's why we're doing these kind of studies. We know that the adult flora can be affected in early childhood. We know that disturbances of the flora can be linked to disease. For some diseases, it's associations. For some diseases, we start to have proof of causality. This means we can go look at your gut flora and move to diagnostics. And I'm predicting that in the future, we will be able to predict the advent of disease. There are long-term effects of antibiotics on the gut flora. And I think we can modulate the flora as a treatment, right? For some of the diseases where the causality is proven, that by changing your gut flora, we can help treating the patients. Some take-home messages. There's now very, very little evidence now, 2-3 papers, that we can find links between the gut flora composition and your brain. Behavior. We see that anxiety, so fear in mice, seems to be linked to the gut flora composition. Explorative behavior, diseases like autism, seem to be linked to gut flora composition. So, and in addition, we see that, for example, mating preferences, these are fruit flies, fruitvliegjes, that the preference of mate, so the male chooses this or that female fruit fly for procreation, is linked to the mixture of bacteria that they are living on the skin of these flies. So this is also behavior. Your mate choice depends on the bacteria, and not by what they look like. I don't see any difference between the different fruit flies, but I guess fruit flies can detect these subtle differences. But apparently, there's something else as well. So when you, smell your partner next time, think of all these bacteria living on the skin producing all these lovely metabolites, right? So, are you really sure you decided what you had for dinner? Because I would not be surprised that it was your gut bacteria pulling at your spinal cord and say, hey, we need some protein down here. I don't do this work alone. There's lots and lots and lots and lots of collaborators all over Europe that we work with, and a lot of funders. And it's mostly these people to the left, the people in the lab, that do all the hard work while I come here and stand on these things. And but I would also like to thank you for your attention, and if you have any questions, I'll be happy to answer them.
Thank you very much for this inspiring and intriguing presentation. And I think there are a lot of questions. I can warn you that last time the former speaker said he never had such an intensive half an hour, even an hour of questioning after giving a presentation here. So who wants to be the first? Front row. Oh, please wait until the microphone is presented.
Graag in het Nederlands. Ik heb 2 vragen. Wat is het effect van het inenten van jonge kinderen? Dus niet alleen antibiotica, maar gewoon alles wat ze tegenwoordig ingespoten krijgen. En ten tweede, wat is het effect op het niet geven van borstvoeding?
Okay, so the question was, what's the effect of vaccination? And the second question was the effect of not breastfeeding. The answer is we don't know. From the studies that I've seen, I don't think there's a big difference in gut flora composition between vaccinated and non-vaccinated individuals. So I don't have much to say on that front. Breastfeeding, on the other hand, is an important determinator. We see children that are breastfed have a very, very different gut flora composition to those that are not breastfed. Again, I think this has to do first with the type of nutrition, because by giving the breast milk, you bring in certain food items into the young intestinal tract, and also you influence the immune system. We know that by breastfeeding, you get a lot of immunity given to you by your mother, and so that also affects the gut flora. So we don't really know from a gut flora perspective whether it is healthier to breastfeed or not breastfeed. We know that there's a difference. I don't have any real recommendations. My children were breastfed, not by me.
Hi, I'm interested in the antibiotic effect that you observed. So you saw that at certain moments you have the people that do not really recover from the antibiotic treatment, right? But it doesn't have to be bad, right? I mean, you can reach a certain alternative stable state in these communities. And but how would you define then if it's bad or not? How could you define the functionality of this community?
It's a fantastic question. You want to work in my lab? The situation is that we don't really know, right? We don't really know whether the antibiotics treatment has ended in a worse or not worse state, right? We only see alternative states. We don't at the moment. When we look at the enterotypes, we have very conflicting results so far. So we see a lot of diseases in that Bacteroides corner, for example. But we also see diseases in the corner where we thought that it was healthy for 1 or 2 studies. Maybe these studies were wrong, maybe we are wrong, you don't know. This is how science develops. At the moment, I cannot judge whether it's healthier or less healthy. What we only know is that these people have altered their thing. Again, this is, for one, I have to say that this was not my study, it was somebody else's study, and also that this was a small number of individuals. These were less than 10 people in that study. So this is topical evidence, and you need large scale studies to really prove this, and to really show what the difference is. The only thing is that the only message that comes out of a study is that different people really react differently, and that antibiotics can have long-term effects. And that's how far we can go right now, I think.
Can I ask a very simple personal question? Should we already freeze our fecal samples during healthy periods in order to recover as quickly as possible after antibiotic use?
Yeah, and send the sample to me as well then.
Oh yeah. But would it be beneficial?
I've said so. I mean, I've been interviewed for a journal recently, and they said, should we do it? And I said yes. I think we don't really know yet, but the way, I mean, then again, the way you freeze it, etc., matters a lot. But that's why I said send the sample to me. We can keep it safe for you.
Thank you. What was the next here?
Actually, Gerber just stole my question. But for another, maybe simple question, would you maybe agree that there's another enterotype, maybe simply the bifido enterotype for babies?
Yeah. Okay. So you're absolutely right. So what I said here is about the adult flora in the Western world. That's where it varies, because the baby composition is very different from the adult flora, and it doesn't fit in these three. So what you say high on bifidobacteria, things like that. What we also see is that, for example, in non-Western world, Native American Indians, black Sub-Saharan Africa, we actually start seeing different types of gut flora that we have not seen before, or that do not really fit into this thing. So the enterotype concept is still evolving. We are not, this is not the end of it, and I'm not gonna, at the moment it's sort of these three, but I'm sure it will evolve in the coming years.
Also, for the people in Africa, they have an extreme Prevotella.
Yeah, exactly. But it is like Prevotella, but even, very far out actually. Yeah.
There's a question back there, and then we'll be over there.
I'd like to ask a question about genetic descent, perhaps.
Mm-hmm.
You said something like, in one of your samples, you had a father and a son, and their flora weren't comparable.
Yeah.
And then you've said some other things about the role that the mother's flora do have on children.
Yeah.
Have you looked at the mother's line to see if perhaps flora are passed down that way?
Yeah, so we haven't. There's been some topical studies. So there seems to be more similarity between a child and the mother flora than a child and some other flora in these studies, right? Very careful again. Monozygotic twins have more similar flora than dizygotic twins have more similar flora than brothers and sisters. There is some genetic effect to some extent, but environment does really matter a lot, and food and other things. There is some genetic effect. There is some what I would then call epigenetic effect, where you have the transplantation from mother to child, which has nothing to do with the genetics, well, sort of indirect, but really by transfer. At the moment, we haven't really quantified really what the relative importance of all these factors is. Also because we don't really know all of the factors that influence the flora composition. Like we've done it here in mice. People are now starting to do this in human, but these are only limited number of studies. So the answer is yes, there is an effect, but we don't really know how much each factor weighs on the final picture. And again, we have not touched here at all on temporal variation. We know that the flora is sort of stable over time, but not really, right? It can alter in a lifetime. Again, there's very little data on that front. We have some scientists that have looked at flora over a course of 10 years for 1 or 2 or 3 individuals. And they've seen that in these 10 years, the flora had shifted sometimes, but for others not. But again, these are very small studies, so we don't really know what the general trend is here.
There's one over. Then in the back.
Wat is er bekend over de luchtwegen als parallel tot het spijsvertering?
Ja, this is a flora. The question about the pulmonary flora, the lung flora. So this is a very interesting topic of discussion. For a very long time, people believed that the lungs were sterile, completely sterile, that there was no bacteria living there. More and more evidence is showing that there's also bacteria living there. I wouldn't say of a resident flora yet, because we don't really know. But again, it's a very controversial topic, because sampling, when you want to sample the airways, is very, very difficult, because you have to go in with a catheter and then take a sample, and then you have to make sure that what you're sampling there is not a contamination from your throat flora and this and that. So it's being investigated, but it's still very, very early days, much more early days than in the gut flora.
Over there, and then we go front here.
And this can take a while.
I was wondering if you have some knowledge about the robustness of the different enterotypes. Let's say if you take antibiotics, that one enterotype might have be less stable than another one.
There's no, again, this has to do with longitudinal studies. There's very, very limited longitudinal studies that have in the past collected fecal material, because nobody was interested in fecal material and everybody was collecting blood and urine, but not this. And so most of these questions can only be answered when the studies that are collecting right now, like ours and many others across, will be able to analyze this. There's not much data out there on flora that has been sampled for more than 2 years or something like that. So the answer is we don't really know. We don't know which type is more stable. I can give you some guesses, but it's nothing proven yet.
We go one up here and then we go in the back.
I have a question about the causality of the changes in the gut microbiota. You show the obese mice, and if you transplant the flora into a sterile mouse, the mouse gets fat. But it would probably not happen if the mouse would have its normal microbiota.
Yeah. Yeah.
And in addition, the second question, would it not be more interesting how this fat mouse got to the point of this bad flora? So if you go to the humans, how do we get the bad flora? We probably don't get it by eating healthy. So what's causality in this?
Yeah, very good question. This particular experiment was indeed done in germ-free mice, and so you could think like, well, what is maybe just only bringing in the flora has an effect already. I think they controlled for this, but I don't know in detail. Now, for example, the diabetes example, there you have people that have a flora. So the only thing that happens there is that they get the intestine is sort of washed out, and by doing that, you don't destroy the previous flora. It's still there, but in lesser amounts. And by bringing in this new flora, then it completely takes over. Also, we see the same in ulcerative colitis and in Crohn's disease. Also, there's also Clostridium difficile infections. There's also a very important example. This is people with very heavy intestinal infection caused by one bug. Fecal transplant has more than 99% success rate in these kind of cases, where also the only thing you do is you just reduce the abundance by trying to wash out the flora or the intestine as much as you can, and by bringing in an ecosystem. So yes, I don't think we have gigantic amounts of proof for causality. I think there are hints. And this is the first. I mean, I'm already happy to give these examples. 2 years ago, I couldn't even give these examples. This is an early research field. We see these first trends. We think there's causality playing, but we don't have proof for all diseases. You're absolutely right.
Still over there. Oh, here is one besides you. Yeah, and then we go.
Hi. We've currently categorized people through geography and age as well, but has your research or any other similar research suggested differences between genders, between people, so male and female? Yeah, a little bit, but it's not a big difference. Usually male and female tend to cluster together in most of the studies. If you dig down to the details, you could find some fine differences which you could attribute to difference in hormone householding, or basically difference in food habits probably, but it's really not the main determinant. That's not really a big effect. I think inflammation, food, and antibiotics, disease, these are much bigger players than gender.
Yeah, there's one more over there, and then.
First of all, thanks a lot for your inspiring talk. I was surprised at one point because you were talking about the negative consequences of antibiotic use, so that your gut flora could change even on a long-term basis. I was confused because you were also talking about like the genetic analysis of bacteria, and that it's just recently been possible actually, and that there's just data for like the last few years. How do you know about this like long-term scale?
Because these are indirect associations. So, for example, these long-term scale effects of antibiotics usage and allergies and eczema and all these kind of things, these were observations purely on phenotype. And it's only recently that people then start to make the link between the antibiotics usage, the gut flora, and the eczema and the allergies. So that the original studies are older studies, and they just make this link. So that's why we don't have data from individuals for like 30, 40, 50 years ago where we had gut flora before the antibiotics usage. We don't have that data, but it's just it is indirect inference.
There's one up here, I think, and then we go to the back.
Maybe a similar question. We know that different people get different side effects from antibiotics, like allergy or diarrhea. But are these? Do you know if there is any correlation between these side effects and the long-term change?
Well, the disturbance of the gut flora can certainly be linked to diarrhea, but I don't think it's much. There's not much research specifically into the side effects of antibiotic usage and then the gut flora. I mean, those studies don't exist as far as I know.
Okay, we go to the back here. Yeah.
If you know there's something wrong, well, you go to a doctor, and normally you don't get far from like IBS or chronic fatigue syndrome. And my question is, in the alternative medicine, you can get these examinations for the stool, and regular doctors, well, I don't know, maybe they don't do anything about it because it's not proven yet. How do you think about people starting using on their own like probiotics and enzymes?
Okay. Yeah. There's a lot of questions in one question there.
Yes, sorry.
So, there's always this fight between tradition, with classical medicine and alternative medicine, and a lot of the classical clinicians they brush away everything that's on that side and vice versa. And there is a lot of things that I also would not agree with on that front. On the other hand, the things of that area are also based on practices that have been passed on from generation to generation. And so one good example here, for example, is the fecal transplant. Fecal transplantation is now a hot area in clinicians studying IBD, etc., etc. Fecal transplant has been used by Bedouins for ages. Fecal transplant is used in veterinary science for ages, where stools are transplanted from one horse to another if that horse had problems. In old Chinese medicine, fecal transplant was described. So in this alternative area, there are practices that are performed. We don't really know the scientific basis for it, but they are performed anyway. Now, of course, this is not the way a classical clinician would work, right? They use this fecal transplant because there was gigantic successes, especially, for example, in Clostridium difficile infections. But they don't like it because they can't control it. You're transplanting something of which you don't really know what it is, right? And so the clinicians would love for us to find out which bugs are in that fecal transplant that actually do the difference, that we can only give those bugs and be very controlled. And I think that's sort of the difference between the two areas. So am I brushing away everything that is alternative medicine? No. Do we have to be careful? Yes. Right? I also get emails from people in alternative medicine that for example, would promote these extreme forms of fasting until your whole intestinal lining comes out. I am very much against this. Right? This is not healthy. But yeah, it is a practice that some people believe is healthy. Chronic fatigue syndrome, IBS, very interesting area. So these are diseases of which certain groups of clinicians would disagree that they are diseases. Right? Yet there's people, if you have a true IBS patient and a true chronic fatigue syndrome patient, they will tell you that this is indeed a serious illness. The problem with these kind of things is that it's very difficult to diagnose. It's very difficult to say what is a chronic fatigue syndrome patient and what is somebody who is depressed and feels tired because they're depressed. And that's a very difficult call to make. And that's why these diseases are always in the gray zone. But yet again, there are studies being performed in CVS, in IBS, on gut flora, and don't think that the science is not looking at it. We ourselves are in a European consortium focusing on irritable bowel syndrome, so it is being investigated. Probiotics, another interesting question. So also there you have a gigantic gray zone, right? You have on the one side the producers of probiotics, Danone, Friesland Campina, all the guys, Yakult, and they have their products and they want to sell their products. The proof for these products is not always there, right? There's a lot of studies that with probiotics that have the end conclusion is there is no difference, there is no result, there is no effect. There are some studies that show results, but that's the minority of studies. So saying that, if I'm as a scientist, honestly saying, do these probiotics, I'm not going to focus on one of them or whatever, but do they affect your health? And then the answer is yes. Sometimes, in certain circumstances, they might produce, but in many cases, it's just some yogurt, right? And it tastes good. And so it's very difficult. And you have many different classes of probiotics. You have these days we have probiotics. We also have prebiotics, where there's food ingredients added. That you eat food ingredients that promote the growth of your gut bacteria. So instead of ingesting bacteria, you ingest food for the bacteria. You have synbiotics. I strongly believe in flora modulation as treatment. And so I'm not wiping away the whole area of pro and prebiotics. The only thing, I think there's a lot more research that needs to be done to find out when it really works and in what situations. Because I think there's a lot of overselling happening at the moment for many of these products.
Maybe I can add a small question from my side. So 2 weeks ago, this audience got the advice not to go for this one strain products, but just go for the simple yogurts. Would you agree with that? The old-fashioned yogurts with mixed strain.
Yes, yes. I think this, and I think this the producers themselves are also moving into that direction, right? There used to be the fact that there was this one-track mind of bifidobacteria, as they were mentioned here. Bifidobacteria, they're healthy, right? And if you increase those, then your product is good. Well, I think we should move away from that. It's too simple a definition. With all these techniques we now know, it's not as simple as that. And these producers, they also know that it's not so simple as that. And so they are also adapting their products. And I think the probiotics will evolve into multi-strain probiotics. And multi-strain plus these prebiotics all mixed together. And I believe that these kind of things will be more effective than single strain. And but these are not much on the market yet. So I would at the moment agree with, raw milk-based yogurt and raw milk-based cheeses is probably the best thing you can do at the moment.
Thank you. There were still questions left. Oh, a lot, I see. We go from there.
I went on an interesting topic here.
Yeah.
Actually, I have a whole different question. Might be a little bit of a strange question, but I'll give it a try. So you mentioned all these kinds of bacteria that you have and the effects. But if you take one group of bacteria, then the phenotype these bacteria have also depends much on the environment. So if you have an environment in which it's hard for them to live in, they will express all different kinds of proteins and in a really easy environment. And I think you touched upon this a little bit in the beginning when you said, okay, we can also measure the products they are making, for instance. Is there more known about this? So people that have the same enterotypes, but that these bacteria are stressed or...
Yeah, very good question. This is an area that is still developing. So I told you about metagenomics. And in metagenomics, what you do is you take all the DNA from the bacteria and you look at the DNA. But you can do things like what people call metatranscriptomics, metaproteomics, metametabolomics. And there you look at the proteins that these bacteria make, the metabolites that these bacteria make, and then you can compare that across individuals. So you don't see what the gut flora or these bacteria, what they can do, but what they are actually doing right now. And then you can do all kinds of interesting intervention studies, right? You can have somebody eat French fries for a week and then monitor the gut flora during... I'm Belgian, I talk about French fries. This is a growing area, let's say. So people have been looking at this. Here's something for the probiotic world. There was a study done by an American lab, sponsored by Danone, and where they looked at the intervention with their product into the gut flora of individuals, and they saw that if you give that product, that did not affect the gut flora composition. Nothing. However, this study looked at the transcriptome, at what products these bacteria were making, and they did see a difference. So the conclusion of the study was the probiotic does not affect the composition of the gut flora, it affects the activity of the gut flora. It's one study, right? I don't make any judgment here, but it's one study that shows that an intervention with a product can change the activity of bacteria. And you will for certainly have that with food items, that if you suddenly start eating a lot of fiber, you'll see different activities. And it's ongoing. We've had studies where we looked at using proteomics at the activity of the ileal, so dunne darm, small intestine flora versus the colon. And there we could see that by looking at the activity, by looking at proteomics, we could see that the enzymes and the products that were being produced by the ileal flora are very different than that of the colonic flora. And that the ileal flora is mostly active in the breakdown and take-up of small, easy, fast sugars, while in the colonic flora you would have slow fermentation of hard-to-break-down fibers. By looking at the activities, not at the composition. So these kind of things are ongoing, but this is still more emerging.
Okay, we'll go to the last. I think here were still 3 questions here.
At the start of your talk, you suggested we could think about this gut flora as an organ in itself. Mm-hmm. And most organs in our body are shaped by natural selection, have been shaped over a long time course.
Yeah.
Do you suggest that we should think about this gut flora also as a trait that's under natural selection? And if so, is there any evidence of relationships between this gut flora and reproduction and survival?
Fantastic question. The answer is I don't know. The answer is I agree completely, but there's not much. I don't think of much research that has been done. I would think that answers to those kind of questions would probably come from the Drosophila field or the fruit fly field or the mouse field much earlier than from the human field. because there's for evolutionary differences you would. I mean, if you want to do this in human, there are some people studying ancient fecal matter, but it's hard to get. There is the Neanderthal flora and things like that. No, fantastic question. I agree. I think it's under selection, but we don't really know. Intriguingly, it's not an answer to your question, but for example, we do see that flora composition is similar across different mammals. So we know that orangutans and gorillas, I think they also have the enterotypes like we have and things like that. So there seems to be some evolutionary pattern, but it was never analyzed in the way you tell.
Was one up front? Yeah, you already had one question, so maybe you can talk afterwards.
So my question comes very close to the previous question. With natural selection, there is also a very big influence of variation on how things progress. And my question is it more healthy to live among people with different enterotypes than your own enterotype? And I can think of two relevant situations. For example, children in daycare versus being raised at home, and elderly living at home versus in a nursing home.
Yeah.
And what is your idea on this?
Well, it depends, and that has not been quantified. We are not mice, so it depends how much of our gut flora we exchange with our people in our surroundings, right? And with children in daycare, I can vividly imagine this exchange happening a lot. I think it's also, again, I think there's actually an adaptive process going on there. I think it's that there's, I mean, this is just a theory, but I think there's the fact that babies, whatever they take, they put in their mouth, I think actually that is a trait for increasing diversity, but on various fronts, not only microbial, but also immunology. In the study, but I don't remember the details, people in the elderly, there was clear difference in gut flora diversity between people that lived still at home and went into elderly homes. There was a big difference. The flora really shifts in that transition. Is it because of exchange of gut flora? I'm not sure. It could also be exchange in dietary habits. It could be, it could be many factors, but and also age-related factors. So yes, very relevant question. The babies, has it been studied? Daycare versus home? I don't know. Could be. I don't know. I think I can imagine it has an effect. The only question is it will be very difficult to disentangle every factor playing there, because it's not only exchange of food, because also children in daycare, they probably get different food than they would get at home. There would be more stress for these children, etc., which also could affect, they would get more diseases like flu and things like that, so which also could, so there are many factors that could influence that thing. But it's a very good question, again.
Well, I don't have a question, just a remark. You come from the country from Tel Uilenspiegel. And you know the story of Tel Uilenspiegel. He was selling profetenkorrels. They were pills made from fecal material. Okay. That's what I wanted to say.
Interesting. I'm gonna use that for some of my future presentations.
That raises a question immediately.
No. The other one raises my question. You said there is a change between elder people living at home and living in the institute, but you did not tell us, is it for better or for worse when they go into... It can be for better if the food is better.
It has to do with dietary habits also. I mean, I think there's several classes of people in both groups. I mean, it's not only the location, right? It's also food habits. If people... You can have people that have, for example, before they went into the elderly house, really were digressing into very limited food habits and very bad habits. Then they would probably benefit from going to elderly houses, while other people that would have a healthier lifestyle would move into a more narrow, would have a... I don't know that it's published. It's only one study actually, but it's I don't know the details anymore. But I think it was not clear cut only this or only that. There were multiple subgroups in these different groups.
Is there somebody left who has a burning question and hasn't been asked yet? I see one here, so we take this.
I'm still here after this.
Okay. So afterwards you can still ask questions.
I have a question regarding body temperature, because there's individual variation in body temperature, and the gut microbiome is of course selected to live in a stable environment, well, relatively stable with body temperature. Do you think individual differences might be related to individual differences in body temperature?
We don't know. We wanted to look into this. When we saw the enterotypes, we also wanted to look at that, and we didn't have the data, so I don't know. I don't recall anything being published in that direction, but it could be. But no information.
Okay, thanks. We still had one in the back, I thought, over there, yeah? Take that one, and then the last one's up front here. Yeah?
Do you know something about the influence of house animals, dogs, cats?
Great question.
Our mother told us.
Yes, that's the one that was still missing. Apparently, yes. So I have a colleague in the US who has studied that, Rob Knight, and he has been looking into this. So pet owners, they do have different floras than non-pet owners, and also this is believed to having to do with being exposed to different types of bacteria from these pets. Then again, one study. I mean, it is almost scary. Every story I tell here, every observation is based on 1 or 2 studies at the moment. But there seems to be a pet effect, beneficial apparently. Yeah. Yeah.
Okay, and if I'm right, there was one question up front here, so we finish with that one. And those of you who have additional questions, I said it will be longer, so you can ask a question.
I was wondering if you know anything about any medicine for any disease, the effectiveness, as how that is dependent on the flora that you have, and if perhaps maybe the screening of the flora that you have can be used for determining what medicine.
Absolutely, fantastic question again. No, seriously, we know that the gut flora metabolizes everything we eat, and so also drugs, so medicinal drugs, are being to some extent metabolized by the gut flora, and there have been 2-3 studies showing that the pharmacokinetics, as people tell, so the concentration of the drug in the bloodstream is also linked to the gut flora composition, because they do affect the routes the drugs are taken, and whether they've been sent for breakdown or whether they've been kept in the bloodstream. So yes, I think it's also a very important emerging field where the gut flora composition might actually determine how people react to certain drugs. Yes. Okay. But again, number of studies is very small.
With this answer, I'd like to close the questionnaire. If you have additional questions, you can come forward. Thank you very much for your presentation. Thank you very much for all the answers. I hope you've been challenged a little bit. You also generated some ideas.
There's lots of specialists here.
Okay. And of course, we hope in years from now that you may even come back and tell us about all the progress and what we should do and not should do. Personally, I will go home with the question, whether it were my human brain cells that drove me here to listen to this, or it were my microbes that want to hear how they should be treated properly. As a token of thanks, we have 2 presents for you. Hope you will enjoy the reading, and once again, thank you very much for your presentation and for coming all over to Groningen. And I'd like to thank all of you for your presence, for your questioning, and hope you'll have time again in 2 weeks from now when Lisette van Lieshout will come here and tell about our parasites that we have in and with us, and if they are for good and if they are for bad. Hope to see you in 2 weeks' time. Have a nice evening.
Yeah, because this is a fairly new field increase the whole system. But if you take a simple one, krijg je iemand die aan zijn dag ja. Ja, dus it takes some simple just like flora that got flora. Thank you very much for interesting.
It's a very strange family because in one generation we have more than like 15 siblings, and only a couple of them have disease, and it's really very dangerous. So I had this idea that we but I think Russia is so because these people are more susceptible to severe infection. And that causes
Yes, the viral is also a coming area. It can be also used to safety needs to study these viruses. It's very tricky. It's very especially for those who basically not to work with.
some viruses only have
Yeah.
some of the single-stranded
But it's also getting so it's the different experts, it's also just getting the viral particles out to sample the correlation is also specialist work. It's also very interesting. It's nice, eh?